Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members in the broader scientific community about topics related to antibody discovery and development. Our expert speakers, Zev Wistotsky and Samantha Jeskonik, will be discussing enhancing antibody developability with AI, a design, make, test, decide approach with us today. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Also, keep an eye out for the 2 polling questions that will be made available during the presentation. Without further ado, I'll turn the show over to our speakers.
Awesome. Thank you, everyone. Thank you, Kristen, for the kind introduction. We appreciate everyone's attention and engagement today. So yeah, as Kristen said, please put your questions in the question section, and we are eager to answer them. Let's jump in. So before we go into the main part of the talk, for those of you who are unfamiliar with Revity Signals, I just wanted to give a little overview. So Revity Signals is the software and informatics division of Revity, and our focus at Revity Signals is developing innovative software solutions. Our mission really and our impact is we aim to assist scientists in creating new medicines, and ultimately with the goal of saving lives through these advancements. So I'd like to start out by talking about some of the key challenges facing pharma and biotech today. And first of those is the increasing complexity of drug discovery research. For those of you obviously are focused on antibody modality drug discovery, but over the past 15 to 20 years, researchers have been working with a broader spectrum of molecules, ranging from the small molecules all the way up to cells and beyond. And these diverse drug modalities are not only harder to represent, but they are also harder to make. They're harder to deliver, and they necessitate really greater collaboration across scientific disciplines. And this means we need better software, or we need better tools to communicate about them, to create them, to track them, etc. And I don't need to tell this group here, but the complexity is further amplified due to the intricate nature of the biology involved. So to manage this complexity, we really need a next generation discovery informatics solution. And as I mentioned, today we'll focus on antibodies, but you really do need a system that potentially can support all of these or many of these modalities, as a lot of times some of them are cross modalities or mixed modalities. So that's why we've deliberately built software to support an end-to-end scientific workflow from the designing, making, testing, and deciding phases of drug discovery. So Revity Signals really stands out by offering software technology that's designed specifically for researchers. We come from kind of the chemical intelligence side, founders of ChemDraw, kind of the gold standard in chemical communication. I'm not going to talk about that today, obviously, but just to mention that. And then we also stand on the shoulders of Spotfire for advanced analytics and visualization to bring that into our full suite of tools. So Signals Notebook, adding the various different experiments, etc. And we're really showcasing today part of SignalsOne, which is kind of that end-to-end piece. I'll stop kind of giving the elevator pitch, but really today's presentation, we're going to talk about antibody developability, furthering what we discussed from our last year's webinar on antibody drug conjugates when we presented with the Antibody Society. So again, before I jump into the meat of the conversation, I just wanted to give us a framework. So the assumptions we're making today are that you understand some basic basic antibody R&D, as well as needing or understanding the need for a software to support those. People are typically not writing antibody structures on paper anymore, if they did at all. So antibody science, as in general, there are various different nuances, but for the most part, people are designing some aspect. They are making these antibodies and reagents for testing. They're then testing them with primary or secondary screens. And then they're deciding, are these candidates, or do these candidates pass muster? Do we need to redo the cycle, et cetera? And again, today we're going to focus expanding on what we talked about last time on the make and test, but more focusing on the design aspect and the deciding aspects. So just again, to remind everyone, we discussed heavily last time the make, test, decide when we showed the ADC talk last year. This is just a reminder that SignalsOne easily enables teams and researchers to do this, showcasing the ADC use case. But again, today we're going to focus on the antibody portion. But I just wanted to remind everyone who is working in multimodality teams that it is possible to work within the same platform for both chemical and biological workflows. All right. And so this is the last slide that I showed from last year, but I think it's important to emphasize kind of where we're going and the dashboard, kind of showing you the end first. So, on the table on the right here, we're looking at a dashboard where you can see things like, molecular structure, various different sequences, and a bunch of different data that's been captured on, in this particular example, ADCs. And the key point here is that we can generate a multi-parameter view of all of that data and that complexity all in kind of one place. And the point of that is when decisions are being made, if we have a zone that we're interested in, right? Each of us, within different organizations are going to have their kind of different rules, business rules, or scientific, insights that they have. And that can be modeled within sort of this green area. We'll call it like the Goldilocks zone, even though it's green, where, it's not too hot, not too cold for antibodies, right? And our goal is to kind of compare against the various different candidates that we'll get to, which of these we want to move forward. These are very expensive, time-intensive decisions that are being made in our organizations. And the important thing here is that we're able to trust the data, we're able to make decisions clearly. And then if we were to go back, 6 months, a year and more, we can kind of intercalate, hey, why did we make this decision? Oh yeah, this is that dashboard we used to do so. This is the information we knew at the time to move that forward. So I just wanted to emphasize this from our last talk, because this builds upon this and helping both the end user scientist get access to tools that will help them make antibodies better and have less liabilities, et cetera. You'll see from our talk, but also the team as a whole to make big decisions on which candidates to move forward. Sam, did you want to interject?
I wanted to mention here that, you're showing the SAR aspect, I think, in the table form in the presentation that you gave last year. But what we're going to show today kind of deviates a little bit from that. It's kind of table stakes. So you can build an SAR table or a sequence activity relationship table and have, here are your different, sequences. Here is your different data points. What we're diving into today are really the workflows that we have built around supporting antibody developability and really focusing on one of those particular modalities that, we support many of in the SignalsOne product.
Yeah, I totally agree. So let's move in. So Sam, I think one of the things I can really appreciate when we're building some of these is we're opinionated and we're using antibody scientists' input to make sure that the tools are focused on that particular workflow. All right, so we're going to just quickly jump into some of the major challenges, and then I'm going to showcase what we're all here to see. So on the design aspect, using that framework I mentioned, one of the big challenges is selecting high affinity hits that later fail to biophysical liabilities. This is kind of where this happens, but the couple others that I want to mention are lack of structural visibility of where the risks lie. We're looking at these kind of flat 2D linear sequences, and we're saying here are all these liabilities, but really kind of need to understand how that is seen in a 3D way. And then the last one for design is inconsistent or manual liability screenings across teams. You have your bioinformaticians who have their own maybe pipelines that they run through to say, yep, here pulling out these liabilities, maybe potentially separate from our end user bench scientists. And wouldn't it be nice if there was kind of a tool or a dashboard that we can kind of all work from or build from that is in a centralized place, and we're all kind of using the same nomenclature or the same definitions. And then from the design standpoint, it's really, there is very little objective bias to triage or borderline candidates, and the difficulty comparing large panels side by side. So this is kind of some of the challenges we'll talk about through our conversation, and we're really excited to engage with you all with questions and others to see if there are other challenges that you face in your how you're going through your antibody developability or screens. All right, and then I know I'm teasing everyone. Let's do one poll question, and then we're going to jump into the dashboard right away.
Okay, so the first poll question, which I will send momentarily, is what is the number one goal you are looking to achieve with antibody developability tools? So your options are identify potential liabilities early in development, compare and select optimal antibody candidates, guide antibody design and engineering efforts, or not sure, still exploring options. And it looks like we have some voting happening. The polls are moving around. I'll give people some time.
It's a pretty close race. And are we able to show the results on the screen?
Let's see. Yeah. Just let me know when you're ready and I can publish them.
I think so.
I think I've seen the movement stop.
All right. Here we go. Wait. Can you see that?
I cannot, but I can tell everybody here while we figure out how to get that on the screen that it is a pretty even split. So there's a two-way tie, right? So the top two options were identifying potential liabilities early in development, and then also comparing and selecting optimal antibody candidates, with the others of guiding design and engineering efforts and still not sure exploring options were also tied for second. This makes me really excited. I think one of the areas that SignalsOne shines, especially in this SAR and decisioning piece, is really the comparison, right? Looking at data sets together, looking at your different antibodies together and selecting which one's best. And really, one of the top options, compare and select optimal antibody candidates, I think aligns really nicely with what we're going to talk about today. Likewise, I believe, because I've seen your deck, Zev, we have a whole section on identifying liabilities early in development. That was the second option that was most selected. So I know at least half of the audience, at least those that respond to the poll, are going to have some answers or points directed particularly at them.
Perfect. That's great. Yeah. I would say I'd be surprised, but I'm not. And I'm excited again, as Sam mentioned. Thank you for your engagement. Let's jump in and see how we can support you in some of those ways. Okay. So here's the antibody developability dashboard. On the right-hand side, you can see this. Let's walk through it. Before I jump into each section, even the best binding antibodies can fail to developability issues like aggregation or poor expression or high viscosity. We all know this. Revity Signals really offers an integrated solution to assess these risks early, to compare sequences side by side, 3D sequence-based liability comparison. And we've gone further to really develop this opinionated antibody developability dashboard. So just quickly to give you the background on this, it's modeled off a recent paper from the Dean Lab at Oxford called Immune Builder, and it's specifically the A-Body Builder 2. I'm sure there will be a lot of questions on this. Happy to answer that. But just to give you the high level, like how this model was created, it was around 250 market antibodies. So these antibodies are considered, good antibodies. They're on the market. They're doing well. They're doing the things that, have spent many years to get there. And so you're calling those kind of the good characteristics. And that's what the model was built off of. And we've not only put that into SignalsOne, but we've even enhanced that, made it better, made it more accessible. Sam will talk a little bit about that. But let me jump into some of what we're going to show today, and then I'll turn it over to Sam for some of the major questions that we get. So the first one is this 3D viewer. It showcases the sequence of interests. We'll talk a little bit about the surface accessibility, really exciting, and potentially being able to bring out the aggregation prone patches. So typically we're only looking at sequences in a kind of a linear fashion, and this really brings things to life. Is able to focus on various different areas, whatever it's just CDR regions that we're interested in, or other potential areas where making things floppy or risky from an antibody building standpoint. The next is what I'm calling the 3D liabilities. So we're automatically being able to flag exposed risk motifs like glycosylation or isomerization as some examples. We'll talk a little bit more in detail about this when we kind of go in through sort of the video demonstrations, but really, allowing you to see what is a true liability. A liability from 2D standpoint is not always a liability in 3D, and vice versa, and we'll see what that looks like. Being able to flag various different, issues in a table summary. So we'll be able to color code various different things that are even outside of the accessibility liabilities. Such as hydrophobicity, charge patches, total CDR length. And then finally, as I mentioned, we have that final, multi-parameter analysis, looking at those 5 different kind of criteria. And, you're able to see where your antibodies live within that, and then select and choose which antibody kind of fits within that Goldilocks zone. But Sam, I know we get some questions on this, and I want everyone to know what those are.
I was going to jump in because you must have heard me complaining or not complaining about this, but reiterating to you, I get like 2 main questions about this anytime we're showing it to prospects or customers, and they're always the same. One is, what actually do I need to get this dashboard started, right? Because if you're looking at what's on the screen, it's pretty involved, right? There's a lot of information. There's the 4 main different sections that you covered, and within each of those sections there's a lot of detail. And sometimes we get the question was like, am I having to manipulate Spotfire? Am I having to have a bunch of IT expertise? Am I having to custom make something from one of the papers out there to generate this? And when we talk about building SignalsOne with workflows in mind and making these opinionated flows easy, what we mean is all you need to provide us is a linear sequence. You put in your linear sequence, you run this antibody developability dashboard, and it outputs everything that you see on the screen. So it's super simple for scientists who maybe aren't informaticians, aren't Spotfire wizards, right? Who just need their data. And then the second question that I get all the time is usually from companies that have an organization that focuses around having some admins or power users who are responsible for kind of setting up these dashboards or these analytics for everyone ahead of time. And the question they get is like, well, I've set this up one way, maybe I've made a slight modification, or I have additional, visualizations that I added from my particular workflow. How can I share this with my end users so they don't break it? And that's also where SignalsOne comes into play, right? Is that full access management control. So what I typically have seen done in my recommendation is for customers that are in that case where they have sort of an admin that creates these dashboards. Again, not hard to make. You put in a sequence and hit go, but sometimes you want some other visualizations in a particular way. You save it as a template. Basically, you save it as read only to everybody in your organization. That means anybody can open it. They can put in their own different sequence to generate this dashboard automatically and any other dependent visualizations, but they can't overwrite that template work. They can save it as their own template, right? Or they can save it as their own analysis. They can export the visualizations. They can interact with everything on that template, but they just can't overwrite your work. And that's really what's given, I think, our customers doing these types of approaches their best path for success.
Yeah, I 100% agree. So putting it in a notebook and then kind of clicking go with various different sequences you can put many in. Let's go ahead and take a look at that. All right. So this is our first short video. Before I jump into this, I just wanted to mention kind of the nuts and bolts. You're looking at a concatenated heavy and light chains. Typically, the input will give you antibody numbering. We can pull out various different regions of interest. So kind of bringing in a naked sequence, and then the system being able to find all your CDR regions, et cetera, et cetera. And then it can break it up into heavy and light chain and start talking about the relative accessible surface area. We'll talk about this in the next section, but I just wanted to mention what you're kind of seeing. And then we're going to focus right now on the 3D visualization. So again, modeled after that A-body 2, the different model that I mentioned previously. And you're going to see the 3D structure, which is the A-body builder 2, and you're going to get 4 different machine learning models from the system that picks up, and it'll pick the best 4 and then produce the images. So this is looking at a zoomed in amino acid here, but again, you can pull it out. So you can see down below, we're selecting the amino acid, and then you can bring out the larger structure. On the right-hand side, you can start selecting different ones. And I think we'll be able to select 2 of them and then, visualize it in different ways that we'd like. So looking at, the liability, well, we'll get to the liability piece here first, in the next, but being able to compare 2 sequences right away. So in this example, we are going to be able to change the, coloration and whatnot. And I'll show you that in just a minute. I think that should still be going. So on the right-hand side, we can actually, select, hey, I want to be able to see these differently, and we can change the cartoon view. We can change colors. So anything that we need visually to see the differences between 2, 3, et cetera, candidates, we can do that in an easy way. Perfect. So we'll move to the next slide, and then I'll kind of show you again another example of where we're going. So this is an example of actually a nanobody, which is really exciting. And again, you'll start to see, really the ease of use of being able to visualize it, move things around. So here's 2 antibodies, will eventually be 3. We can look at them side by side. We can tile them and make sure that each of all of the data that you're kind of like trying to visualize, hey, this loop over here is, causing us issues, or when we do, mutation analysis, what's going on? Let's compare that. Let's look at that in the 3D way. So I'll let this video play. And then furthermore, I'll let Sam interject in just a minute, but this molecular viewer is obviously fit for purpose for our antibodies, but also expands. So you can start to see, the amino acid chain and the loops for the antibody itself, and then we can start to change what that looks like. If you wanted to see the various different elements and need to visualize it in a different way, you have that at your fingertips. But Sam,
yeah, I'd say that.
Would you talk a little more about that?
Yeah, absolutely. So I'd say this is actually one of the more underrated features in SignalsOne, and it's actually been around for quite some time. So the antibody dashboard leverages this as one of the pieces that it includes when you generate it. But it also is available sort of as a standalone component. So if you're not looking at antibodies and you're just looking at like a peptide sequence, you can certainly use the biomolecule viewer with it. You'll talk in a bit, I think, about how the structures get in there.
Yeah.
But say you have a PDB structure, right, and you want to be able to look at it within Signals, you can read that PDB structure here, generate it, interact with it, and all the different colorizations and modeling that you've seen. You'll see at the bottom of the screen too, right? What you see there is a sequence that's annotated, and you can even, not just pick a single amino acid to zoom in on them, but if you want to select a particular region, right, a set of amino acids that you're interested in, you can highlight those, you can annotate them, and then you can zoom in on those as well. So this has been around in the product for some time now, and I think it's rather underutilized. And I'm really thrilled to see it in the antibody dashboard. I think we're going to get a lot more usability on it there. And if you are a SignalsOne customer listening, I encourage you, go check it out. You actually have this today.
Yep. Thank you, Sam. All right, I think I showed that already. So moving to kind of what I think is one of the really premier things that we're talking about today is this idea of 3D antibody liability or sequence liability. So automatically being able to flag risk motifs, as I mentioned, glycosylation or isomerization, and this idea of structure-based sequence liabilities, based on the accessibility. So again, you're going to see this visual of these sequences, these liabilities being called out. So typically, you have many different antibodies you're making, and you want to just kind of compare them for a various different region, and then look at the liabilities within them. And furthermore, what you'll see in this video, the short one, is we'll be able to do that very quickly, but moreover in the 3D version. So in this short version, in this short example, I'm showing you some CDR regions to choose from, just to give you that we can kind of like call those out and focus on those. And then you can kind of look for liabilities here. Before I go to the next video, as this will stop soon, I wanted to call out the various different colorations here, just to kind of give you an overview. So what we're looking at is the in this bottom middle table, the relative surface availability, where we've colored this any color you want, but in orange you kind of give a range of which of these amino acids are how accessible they are, right? When we're thinking about liabilities, it's not just this 2D structure. We need to really look at a 3D structure to understand, is this going to interact with something at high concentrations, et cetera. And so what you'll see in the next video is sort of the calculations of that and how to bin those. But you can quickly call out which of these have potential accessibilities, and then which will be called out in sort of that red, where there's a liability and it's accessible within the 3D structure. So let's go ahead and take a look at that. Next, here we go. Wait, did I go too far? Sorry, yeah, I did. Okay, here we go. So in this example, you'll see again, we're starting with those sequences. I'll pause here once it kind of gets to the data functions, just to talk through this a little bit. So as I mentioned, we're talking about relative accessible surface area. So I'll use RASA as the acronym. But the point is that, being able to see this in 3D is really, really important. So again, you're using the concatenated heavy and light chains. You'll have maybe a column for PDB structures. Right now, we're using a kind of a standard 7.5 cutoff. This is based on those publications that we talked about. But you can change this. This can be, increased or decreased based on some, internal business or scientific rules that you have. And as I mentioned, here are those colors and the gradients. So from 0 to 100%, you can again modify this to make sure it calls out to the kind of GUI that you care about, the interface that you care about. So let's go ahead and see that in motion. So once that's done, we're taking our sequence. Again, we can visualize it in various different ways. And the goal here is to kind of compare two different sequences, or obviously more than two different sequences, for various different liabilities. So on the right, you can select the other antibodies to click through. And then you're going to see here, as I scroll over this, you'll see it'll showcase the individual antibody metrics. So you can see the calculations of each of these, 0% for this particular amino acid. And then for the next one, it might be much higher than that as we scroll over. I want to emphasize this point. I think one of the things to take away from this is, I know this seems a little more manual. We're kind of comparing one off and two off, but you're really able to do this at scale, and especially for the bench scientist who isn't a bioinformatician, who doesn't have the the various different algorithms to kind of, you maybe would run this outside of Signals typically. You're able to do that right here, all in one go.
That's actually one of the benefits, Zev, of the A-body 2 kind of background behind this, is it's so much faster. I think it's something like 100-fold faster. We can fact check me on the paper, compared to AlphaFold 2. So yeah, quite impressive.
Absolutely. All right, so we will move on for time. And, the last one that I wanted to show is, we're really empowering scientists who, again, may not have the bioinformatics background, it's not their day job. You get those insights faster. So let me go ahead and let the video speak. Again, just showing the sequences here. You can see the numbering a little better. And then as we move through, you'll see again how we can visualize a true, liability that is going to cause issues later down the road. So you'll see this antibody here has a liability, and you can see this red circle, which we'll scroll over, actually is going to be a potential problem, as we develop this. And still showing you some of those RASA calculations for each of these. And then being able to compare and pick another antibody that doesn't have that issue, right? So that's the point, is we're able to quickly say, okay, hey, I'm flagging a liability. I know that's going to be a problem. So instead of just like going about my day and building it and then finding out that later down the road, it has an issue, and you can see that how accessible this is too. So that's quite accessible. We can now take it out, look at some other antibodies that we have that we were working on from a sequence standpoint, compare it and see, either let's make a different antibody with a different amino acid, or let's see if we already have a different antibody that doesn't have that C. So you can see right here very clearly that is missing the liability, which is great for us, right? That's going to help us, kind of make that decision a lot easier. Okay, so moving on for time. Let's go to our last poll, and then we'll go through the final dashboards and open it up for questions.
Okay, poll question 2 I've just sent is, what tools is your organization currently using for antibody developability assessment? And our options are commercial antibody developability software, in-house custom tools or scripts, a combination of multiple tools and manual processes, or not currently using dedicated antibody developability tools.
Well, that poll comes in, Zev. I think one thing I want to comment on is, we talk a lot about this being plug and play, right? You don't really need a lot of background. I don't want that to overshadow the fact that if you have experience, right, if you do want to modify things, you can. And there's different levels of that too. So for instance, we talk about there's the ability to do automatic antibody numbering, to label the CDR regions. There's options in the interface to select, what is your labeling scheme? Is it IMGT or Cabot? So there's levels of how much interaction you want, all the way to, you have something super proprietary that you want to bring in, you could do that as well. So we try to cover all pieces of that spectrum.
Yep.
And I think I see the movement on the poll slowing down now. I'm not sure if we can show it on the screen, but I will kind of go over the results if not. By far, the biggest winner here was a combination of multiple tools and manual processes, and almost very few people aren't using any dedicated developability tools. That makes total sense to me, right? I think a lot of these things start out with an in-house script that then you look for its compatibility with other software out there. There's different platforms, SignalsOne included, that includes kind of these different pieces of the capabilities. I think the standalone piece of SignalsOne is that it really covers that end-to-end platform, right? It covers multiple pieces, from doing SAR analysis to the access control to generating a sequence or a structure prediction from a sequence and calculating liabilities.
Yeah, that's a good call. I mean, I think it talks to also potentially some of the security stuff. There's all these great fancy tools out there, but I'm sending a proprietary sequence, or I'm doing something that, really probably shouldn't go outside of our organization. And just to confirm, using SignalsOne, and this particularly is run locally, so it's not something that's open up, to the world when using, potentially tools like AlphaFold or things like that. All right, let's jump in. We have a couple more slides and videos to showcase. So talking about the liability and flag table summary. So again, I already kind of mentioned the various different areas that kind of we think of as, and from the publication of what you want to look at, but we make that easily into a tabular view. You'll see, various different colors here to flag for you. And it's not just like sequence liabilities. It has to do with, total CDR length. So for example, I think for various different region, if it's too long, it's shown that the antibody is less stable. So things like that we can all pull in and basically give you something to kind of smash your antibodies against as a kind of rubric, if we could call it, or maybe add your to a rubric that you already have, as you mentioned, using a mixture of tools in-house. So let's go ahead and take a look at that. So this again is that dashboard. It's zoomed in this case, so you can zoom in on each of those different sections. You don't have to look at the whole dashboard as itself. You can see they're categorized as sequence liabilities and also developability metrics. So from the publication, you can have other alerts, again, in various different ways to say, hey, this antibody has an alert. And then what we'll show in just a second is a couple antibodies that don't have those. So when I select them, those might be a better fit for us along with the liability. So I have no yellow alerts here, which could be, like a good thing for me. Oops, I think this is playing the same one. Okay, and this is kind of the whole point of everything. So at the end of the day, we're getting to this multi-parameter analysis. As we'll call it, the Goldilocks zones. So, you have a bunch of different antibodies here. So you can see your distribution. You have kind of your red zone, your yellow zone, your Goldilocks zone, and you can select and again see each antibody as it goes through these parameters or experiments when you get to that point on how each one compares. So you're able to compare directly. And I think this is really like the candidate selection, or do we need to go back from a design standpoint? Do we need to go back, from a decide standpoint, do we need to go back and start over? Or, are we able to move this forward? And we feel very strongly about this. These are very expensive decisions, as I've mentioned. So I don't want to take these lightly, but to try to give you the tools, your organization or you the tools to be like, yeah, we're going to make this decision, multi-team, whatever. And we feel really strongly from a data trust standpoint.
And even from our polls that's what kind of people, the bulk of people were asking about, right? They want to compare to antibody candidates. And I think this is a great kind of holistic view of how you could do that.
Yeah. All right. So I'm gonna summarize and then send us to questions. I hope there's a lot. I hope we piqued your interest. So, what is the take-home message from this presentation? As I mentioned, we have really beautiful 3D viewer maps to map the sequence liability against the structure. So it's really bringing that liability to the 3D realm and really being able to play with that, intercalate it, select the area of interest, zoom in, zoom out, and compare many at the same time. We can, flag the various different liabilities here and in the calculations that you care about. We give you kind of that out of the box based on best, quote unquote, best practice or what's being used in the field, but you can modify that and change things to meet your particular needs. So standardizing that, flagging the hydrophobicity, charge asymmetry, CDR lengths, all in one place. And then again, finally, kind of to that cross multi-parameter liability table for comparison against many variants. So, to bring this all together within SignalsOne, it's really a unified platform that reduces your task switching and streamlines decision making. We talked about the design, make, test, decide framework that we can apply to antibodies as well as antibody drug conjugates. And finally, I'll just mention reducing that time sink between your bioinformatician teams, integrating predictive analytics into a scientist-friendly platform. So, I think Sam mentioned it, but I just want to emphasize, using resources to kind of create one-off dashboards or run analysis kind of ad hoc is really not a great use of resources or people's time. And it's just very challenging. So putting this in the hands of the scientists as early on in the process and kind of educating them to figure out how to use it and be able to intercalate business rules or scientific rules in here on top of this. If you want to modify, you have that ability. But Sam, let me turn it over to you for any final comments.
Yeah, I mean, one piece I like to emphasize is you're looking at a small slice, shockingly, given everything that you've presented of SignalsOne, right? So we've talked about the antibody developability dashboard here. That's a tongue twister. We've talked about the ADC and the ability to do it like a standard SAR table in the last presentation, really focusing on the biologics. In your earliest slide, you mentioned, right, that we're actually built on top of ChemDraw. So we obviously handle all of these around small molecules as well. So while we focus on, I think the design piece primarily today, some of the design, the make and the test piece are all captured in SignalsOne today. And
Yep.
just the way that we've been opinionated or tried to get across the point that we are opinionated in how we've set up this antibody dashboard, other areas of Signals are also set up in that way, right? So it's these opinionated workflows that are able to get you to your answers faster.
Yep. We're going to turn it over to questions. Before I do that, I just wanted to mention this QR code will take you to the SignalsOne page if you want to learn more about that. But we're really here right now. So we'd love to hear your questions. And I'll leave this slide up, hopefully for eye candy. But yeah, Kristen, we'll turn it over to you. Thank you again for your attention, and we'll take questions.
Great. Thank you so much to both of you for this wonderful presentation. And I just want to remind the audience that you can add questions to the Q&A box in the chat, and I will read them out loud. So I'll get started with what we have right now. Can this modeling predict how the antibody may recognize the antigen of interest?
So we don't do that quite yet. So that would be a bit more manual, right? You're given the 3D structure. You could also bring in, say, the 3D structure of your antigen. You can compare them and see how they might fit together, but we don't have a prediction on that yet. That said, many of our ideas for how we build these softwares and the pieces therein come from user requests and user feedback. So if that's something that we hear a lot of feedback on, that's something we would be interested in pursuing.
Yeah, it's a great call. We're not doing sort of like that binding, direct binding, things like that. But we're actively in development of this, and we'd love to hear thoughts. Critiques, beat this up, tell us what you hate, tell us what you love, and then we can kind of figure that out. Otherwise, we wouldn't want to build things that people wouldn't use.
Next question, are the 3D structures predictions generated from primary sequence? If so, what system are you using to generate these predictions?
Yeah, so they're generated from a primary sequence, looking at the concatenation of a heavy chain and light chain, and they are calculated with Antibody 2, which has some modification to it that our developers who are actually watching, so I hope we're doing them justice on the line, have implemented into that to model to kind of make it more efficient, address some inconsistencies.
Yeah, from the paper. I'll just add
the
on.
Raymond paper, right?
Yeah, exactly. So I'll just add on that it's run with 4 different models within that machine learning algorithm, and you can kind of pick it picks the best model, the one that basically is most accurate. We have done our own internal testing though for antibodies outside of this training set, and just to confirm that this is run locally, so this isn't being sent out. We're using that as the build, but it's not being sent out, so it's all local within SignalsOne.
Can you visualize non-antibodies?
Yeah, absolutely. So this dashboard, so in full transparency, the antibody developability dashboard is really around antibody development. We have one coming out for nanobodies, but the sort of the one click and then get your antibody structure prediction, the liabilities marked, the multiparameter optimization, that's focused on antibodies. That said, there are pieces in there like the 3D molecule viewer that can absolutely just be used on regular protein sequences. Not really structure prediction there, but you could bring in a PDB file, right? And then interact with it with the sequence down below, marking different amino acids, changing the view to best fit your kind of visualization needs.
Next, what is the throughput of this workflow? Let's say if I want to analyze more than 10,000 different antibody sequences obtained with NGS, is that possible?
Yeah, it's a great question. So I would say that the dashboard does about 10 sequences in like a couple of minutes, so you can extrapolate up from there. I would not use this dashboard for a 10,000 antibody throughput. If we look back, if we think back to the diagram that Zev threw up at the beginning, this dashboard's really at the border between that design and decide piece. So it's not so early on in NGS where you're just getting a ton of sequences that you may or may not be interested in, and then trying to structure predict all of them there. This is you've already kind of done that process. You've narrowed it down to, I don't know, maybe a couple hundred antibody sequences that you think you want to move forward with, you want to know more about. That's really what this dash was intended for. That said, right, there is a make piece of the SignalsOne, and we're doing more to address sort of those early on stages of kind of biologic development.
Yeah, the only thing I'll add on is it's about 20 to 30 seconds per antibody, so it's extremely efficient. So just to let you know, on this particular dashboard, which is around 250, 240-ish antibody sequences that were put in, it took about 40 to 45 minutes from what our the people who built this mentioned. So it's relatively fast, but to Sam's point again, you probably could do 10,000. Let us know. We can try to scale that up, but really it might be better to understand, what your why is behind your question. But just to give you a sense of speed and scale. Yeah.
Yeah, I think there's different pieces too happening here, Zev, where if you can, I think that time you gave, the 45, 90 minutes, right, is really the dashboard in its entirety.
Yeah.
All of the pieces of that dashboard are also available to run separately. And so if you wanted to just run the structure prediction, it's more maybe like between 10 and 20, 10, 15 seconds of structure.
Yeah, we can make that.
Yeah.
100%. Great question.
Great. So next, what types of sequence liabilities does the tool identify?
Yeah, I'll take this one. So it's 8 known. So we can put that in the chat, but basically aspartic acid isomerization, asparagine deamination. We can copy and paste those in the chat. But yeah, we have kind of 8 that have been published from the paper, and typically from things like as you get into high concentration of the antibody sitting in a vial or something like that, like, is that going to cause issues? So I would say it's typically not all of these, but like a lot of these liabilities aren't high priority on the biologist's mind. Like when I think about myself as a scientist, when I was in the lab, I'm not really thinking about like how to make this antibody stable necessarily, but it allows us to bring in those business rules and development rules, which are really nice because it does matter from the whole thing. First, we want to make sure the antibody binds. Then we want to make sure it has some characteristics that we want, velocity and stuff like that. How quickly it binds and lets go, gets out of the body, and then now we need to figure out how to make it stable for a long periods of time, and so forth and so on. So yeah, 8 of those right now, and I'm sure if you have your own proprietary, we'd love to talk to you about what that may look like if you wanted to add that.
Thank you so much. So we have more of a comment, and then there's another question. Antibody Builder 2 as a part of Immune Builder package from the Dean Lab at Oxford Protein Informatics Group. That's what the comment.
Just a comment from
yes,
Antibody Builder.
Yeah. Antibody Builder 2. That's correct. Yeah, and from my understanding, when I talked with one of the developers who's actively doing this, they're already working on Antibody Builder 3. So this is happening. We're continuing to make strides, but I think part of this presentation we really wanted to showcase what we've done, how this is, kind of try to get this into people's hands, or get people to kind of beat it up a little bit, ask questions, and see where we want to go from there. So yeah, thank you for that comment.
And to follow up on that, another question, as I understand it, Antibody Builder 2 is a much faster alternative to AlphaFold that performs at least as well in many key metrics, but does not significantly improve the prediction accuracy, which is known to be highly variable for AlphaFold predicted antibody structures. This is a fundamental challenge due to the way that these prediction algorithms use evolutionary information, residue co-evolution, in their predictions, and the fact that this information cannot exist for antibodies. How is your team getting around this big challenge?
I think we're getting around it in the same way that other scientists are, right? We're keeping aware of the literature that's coming out there, and we're assessing and revising, the strategy as it comes up. Like I said, the reason that we went with one of the reasons we went with Antibody 2 here, Antibody Builder 2, was just its increased performance. It does have a slight improvement in the resolution. but your criticism is, valid, and we're interested to see what the field comes out with. We're interested to see how much of a concern this is among our adopters and where we should invest our efforts next.
Great. Okay, just a few more questions here. Um, can multiple users collaborate on the same dashboard and annotations?
Yeah, absolutely. So that's part of the SignalsOne feature. You build this dashboard, right? Like I said, you have the approach of you could make it a template, and then everybody can work on that same template and save it as their own thing and not overwrite it. Or you could have a template that or not so much a template, but you save it as something that can be worked on and overridden by other users. It's really up to you and your organization how you best want to work. In addition, all these visualizations here are easily sent immediately from this kind of more Spotfire interface directly into, say, an Inventa analysis inside Signals, where you can then actually add, draw boxes, add little annotations right then and there.
Yeah, nothing to add. That's yep, perfect.
Great. Okay, just one more call for the audience if anyone else has any additional questions. Otherwise, I'll read. Oh, here comes another one. Can we bring our own internal assay data into the dashboard?
Yeah, absolutely. I love that question. So there's different ways that you could do that. So the SignalsOne does include a data standardization layer behind it. So a way to manage and publish your assay data. You can bring it in that way, and then add that assay data into an SAR table, aggregate it in different mechanisms, even add it to your MPO, right? That multi-parameter optimization graph. So absolutely. And in fact, that's how we see the larger picture of this being used, right? The antibody dashboard is bridging that kind of piece between the build and decide, but what's going to help you decide more than just maybe the liabilities of this antibody is how well is it performing in these different assays. And so of course we're set up to ingest that assay data and then use it right alongside this developability data as well.
Yeah, the only thing I'll add, and I think Sam summarized it well, is let's say, for example, and I think we even had a question about this, you want to add your own liabilities, or you want to add your own sequence interests, like areas that you want to keep or you want to flag. That all can be, thought about and done in bulk, so we can map those as needed. But yeah, very, very accessible, and again, love to hear from you. What are you looking for? Let's talk more, because we are in active development.
Just a couple more. How do users typically incorporate this analysis into their antibody optimization cycles?
Yep, I'll take that. So I'll just simply, this tool really starts to bridge the gap, as I mentioned, between kind of that design and a little bit of design and decide. So really focused on the decide angle, what are we doing? How are we moving forward? But as you can see, you can start to see some of that design aspects, like, oh, we want to create an antibody that has this amino acid in this location or not, and that comparison kind of so kind of bridging that gap between those two major sections within your decision.
Great. And then the last thing is not a question, but just a comment. Thanks for a great presentation. So
thank you for the great questions.
Yeah, great questions, great engagement. I really appreciate everyone's time.
Amazing. Okay, so that is going to wrap up this for the day. If you just give me 1 second. Thank you again for this insightful presentation, Zeb and Sam. The on-demand version of this webinar will be available really soon, and a link will be sent by email to everyone who registered. Feel free to watch this or any of our other on-demand webinars when it's convenient for you on our YouTube channel. And thank you and have a great rest of your day.